Evolution is a detective story written in stone, but some chapters remain maddeningly incomplete. Scattered across ancient rock formations lie the fossilized remains of creatures so alien, so fundamentally bizarre, that even after decades of study, paleontologists still argue about what they were, how they lived, and where they fit in the grand tree of life. These aren't the famous dinosaurs or saber-toothed cats whose stories have been told 1,000 times. These are the true enigmas, organisms that defied classification, broke every biological rule, and vanished without explanation. They thrived in worlds we can barely imagine, built bodies that make no evolutionary sense, and left behind fossils that read like riddles carved in ancient stone. Some looked like living quilts drifting across primordial seafloors. Others resembled armored worms studded with crystalline spines. There were creatures with 5 eyes, spiral shells that grew in mathematically impossible patterns, and animals that seemed to blend characteristics from multiple kingdoms of life. Scientists have given them names, measured their bones, and mapped their distributions. But the fundamental questions remain unanswered. Were they animals? Plants? Something in between? How did they feed, reproduce, or sense their environment? And why, after ruling their ecosystems for millions of years, did they disappear so completely that nothing like them ever evolved again? This is the story of evolution's greatest mysteries, the creatures that science still cannot explain, the fossils that refuse to give up their secrets, and the prehistoric experiments that remain locked in geological vaults, waiting for someone to finally crack the code. The fractal enigma, rangeomorph body plans. The Ediacaran seafloor was an alien landscape, and nothing walking through it was more incomprehensible than the rangeomorphs. These organisms appeared between 575 and 541 million years ago, tens of millions of years before anything with eyes, mouths, or the ability to move under its own power. At first glance, fossilized rangeomorphs look like ferns pressed into stone, or perhaps colonial organisms building intricate branching structures. But the closer scientists look, the stranger they become. Their bodies weren't made of cells arranged in tissues and organs like modern animals. Instead, they were constructed from repeating fractal patterns, structures that looked the same at every scale, like a feather made of smaller feathers, which were themselves made of even tinier feathers. No living organism uses this body plan. Not plants, not animals, not fungi. The fractal architecture allowed rangeomorphs to maximize surface area without increasing volume, which theoretically helped them absorb nutrients from seawater. But how did this absorption work? Modern animals have digestive systems with specialized cells, enzymes, and membranes designed to break down and transport nutrients. Rangeomorphs had none of these structures. Their bodies were essentially quilted surfaces with no internal organs, no circulatory system, and no obvious mechanism for processing food. Fractofusus, one of the most studied rangeomorphs, adds another layer of mystery. Fossil beds in Newfoundland preserve entire populations frozen in time, showing both large adults and tiny juveniles clustered around them. Analysis reveals that Fractofusus reproduced in 2 completely different ways simultaneously. Some individuals spread by releasing waterborne propagules, tiny structures that drifted away and settled elsewhere. Others grew clones that budded directly from the parent organism, creating dense clusters. This dual reproductive strategy is sophisticated, requiring coordinated developmental programs that shouldn't exist in organisms this ancient and this simple. The evolutionary position of rangeomorphs remains hotly contested. Some paleontologists argue they were stem animals, representing the earliest experiments in multicellular life before the animal body plan crystallized. Others propose they were something entirely different, perhaps a separate kingdom of life that emerged, flourished, and went extinct without leaving any living descendants. Chemical analysis of rangeomorph fossils shows traces of organic compounds, but nothing definitive enough to place them firmly in any modern group. What makes rangeomorphs especially puzzling is their sudden disappearance. They dominated Ediacaran ecosystems, covering vast areas of the seafloor in dense forests of fronds. Then, as the Cambrian explosion introduced mobile animals with muscles, guts, and predatory behaviours, rangeomorphs vanished completely. No transitional forms exist. No intermediate species bridge the gap between rangeomorphs and anything that came after. They simply ended, leaving behind only impressions in ancient mud. The microscopic structure of rangeomorph fossils offers few clues. Unlike modern animals, which leave behind clear cellular imprints, rangeomorph fossils show only smooth, undifferentiated surfaces. Some researchers interpret this as evidence that rangeomorphs lacked individual cells altogether, existing instead as giant syncytial organisms, single cells with multiple nuclei sharing one continuous cytoplasm. This arrangement is seen in some fungi and slime molds, but scaling it to metre-long organisms creates structural problems that no known organism has solved. Modern imaging techniques have revealed intricate details, growth patterns, branching algorithms, and spatial distributions, but the fundamental questions remain unanswered. Were rangeomorphs animals? Were they alive in the way we understand life? Did they have genomes, reproduce sexually, or respond to environmental pressures? Every new fossil adds data, but no clarity. Rangeomorphs remain evolution's fractal riddle, beautiful in their complexity, but impossible to solve with the tools we have. They represent a chapter of life's history, written in a language we've forgotten how to read. The symmetry problem. Tribrachidium's impossible geometry. Tribrachidium heraldicum is a fossil that shouldn't exist. This small, disc-shaped organism lived during the late Ediacaran period, roughly 555 million years ago, on ancient seafloors that would eventually become the Flinders Ranges of Australia. At first glance, it resembles a three-petaled flower pressed into stone, or perhaps an ornate pie chart with three equal sections. But this simple appearance masks a profound biological mystery. Tribrachidium has threefold radial symmetry, a body plan that appears nowhere else in the animal kingdom. Symmetry in biology isn't arbitrary. It reflects fundamental developmental pathways and evolutionary constraints. Most animals exhibit bilateral symmetry, with mirror image left and right sides. This pattern dominates life on Earth, from worms to whales, because it evolved early and proved incredibly versatile. Some animals, like jellyfish and sea urchins, use radial symmetry with 4, 5, 6, or more repeated sections. But threefold symmetry? It doesn't exist in any living animal. Plants occasionally show three-part patterns, but their developmental mechanisms are completely different. Tribrachidium's triradial design stands alone, a geometric anomaly frozen in stone. The creature itself was small, typically between 1 and 5 centimetres across. Its body was divided into 3 curved arms spiralling outward from a central point, creating a pinwheel pattern. The arms had raised ridges and subtle texturing, suggesting they were more than simple folds. They were structured features with specific functions. But what functions? Fossil evidence provides no clear answer. There are no obvious feeding structures, no mouth, no apparent way to capture or process food. The underside of Tribrachidium fossils is smooth and featureless, offering no clues about how the organism attached to the seafloor or whether it could move. Some researchers propose that Tribrachidium was a passive filter feeder, using its 3 arms to create vortices that channeled food particles toward a central mouth. Computer modelling of water flow around 3-armed structures shows this is theoretically possible. The geometry could generate feeding currents without requiring cilia or active pumping. But no mouth has ever been found in Tribrachidium fossils. Other scientists suggest the arms were photosynthetic surfaces, positioning symbiotic algae to capture sunlight. Yet Tribrachidium fossils appear in deep water deposits where light penetration would have been minimal. The developmental origins of threefold symmetry pose an even deeper puzzle. In modern animals, body symmetry is controlled by specific gene networks, homeobox genes, and signalling pathways that establish body axes during early embryonic development. These genetic systems are highly conserved, meaning they've remained similar across hundreds of millions of years of evolution. Bilateral symmetry is governed by one set of genes, radial symmetry by another. But threefold symmetry, there's no known genetic toolkit for building it. Either Tribrachidium used an entirely different developmental system that later went extinct, or it represents a brief evolutionary experiment that was never repeated. Tribrachidium's position in the tree of life remains completely uncertain. Early hypotheses suggested it might be an early echinoderm, related to starfish and sea urchins. But echinoderms, despite their radial adult forms, develop from bilateral larvae and show unmistakable five-fold symmetry. Tribrachidium has no connection to this pattern. Other researchers proposed links to cnidarians, the group including jellyfish and corals, which often show radial symmetry. But cnidarians typically exhibit four-fold or six-fold patterns, and their body construction is fundamentally different from Tribrachidium's solid disc-like form. Recent phylogenetic analyses using morphological data have only deepened the confusion. Tribrachidium shares no clear characteristics with any known animal phylum. It has no bilateral features, no obvious front or back, no signs of segmentation or repeated body parts beyond its three arms. Some paleontologists now suggest it represents a completely extinct lineage, a branch of multicellular life that explored triradial symmetry before disappearing without descendants. This would make Tribrachidium a true evolutionary orphan, a body plan that existed once and never again. The ecological context provides few additional clues. Tribrachidium lived alongside other Ediacaran enigmas like Dickinsonia and Kimberella in shallow marine environments. Fossil assemblages show multiple individuals clustered together, suggesting they weren't solitary. But were they colonial organisms budding from common ancestors, or independent animals that simply preferred similar habitats? Their preservation in fine-grained sediments indicates they were soft-bodied, lacking shells or mineralized parts that would have aided fossilization. The fact that we have any fossils at all is remarkable. Most soft-bodied organisms leave no trace. What makes Tribrachidium especially maddening is its clarity. Unlike many Ediacaran fossils, which are fragmentary or poorly preserved, Tribrachidium specimens show crisp detail. The threefold pattern is unmistakable, repeated across dozens of fossils from multiple locations. This isn't a preservational artifact or a misidentified trace fossil. Tribrachidium was a real organism with a real, reproducible body plan. Yet that body plan makes no biological sense by modern standards. Perhaps that's why it disappeared. As the Ediacaran gave way to the Cambrian, mobile predators evolved, ecological competition intensified, and only the most adaptable body plans survived. Bilateral symmetry won because it allowed for specialized front and back ends, heads with sensory organs, tails for propulsion, directed movement toward food, and away from danger. Radial symmetry persisted in organisms like jellyfish that drifted or sat in one place, where having no front or back was advantageous. But threefold symmetry, it offered no clear benefits and significant constraints. Evolution abandoned it completely. Tribrachidium vanished from the fossil record at the end of the Ediacaran, leaving no descendants and no clear explanation for its bizarre geometry. It remains one of paleontology's most beautiful mysteries, a perfectly preserved fossil of a body plan that shouldn't exist, couldn't be maintained, and was never attempted again. The classification nightmare, Opabinia's 5 eyes. The Burgess Shale has yielded many strange creatures, but few have baffled scientists as thoroughly as Opabinia regalis. This small arthropod-like animal, roughly 7 centimeters long, lived 508 million years ago in the Cambrian seas. When paleontologists first reconstructed Opabinia in the 1970s and presented it at a scientific conference, the audience reportedly burst into laughter. The reconstruction seemed too absurd to be real. 5 eyes, a long flexible proboscis ending in a grasping claw, backward-facing mouth, and a body covered in overlapping lobes that didn't match any known arthropod pattern. But the fossils were genuine, and the more scientists studied them, the more bizarre Opabinia became. The 5 eyes are Opabinia's most iconic feature, arranged across the top of its head like searchlights. 2 large eyes sat on stalks, providing elevated vision, while 3 smaller eyes clustered around them. This configuration is unique in the animal kingdom. No modern arthropod has 5 eyes arranged this way. Most arthropods have 2 compound eyes, sometimes supplemented by simple ocelli for light detection. But 5 sophisticated eyes with apparent depth perception? It suggests a visual system far more complex than anything seen in Cambrian contemporaries. The function of this eye arrangement remains speculative. Some researchers propose that the stalked eyes provided 3-dimensional vision for hunting, while the additional eyes monitored for predators approaching from different angles. Others suggest the multiple eyes allowed Opabinia to see in murky water conditions, where single eye systems would fail. The eye placement indicates Opabinia lived and hunted near the seafloor, using its elevated vision to spot prey above while remaining low to the ground. But why 5 specifically? Why not 4 or 6? The developmental genetics required to produce 5 eyes with precise spacing and coordination would be extraordinarily complex. The proboscis presents an even greater puzzle. Extending from the head like an elephant's trunk, this flexible appendage measured nearly 1/3 of Opabinia's body length. At its tip was a grasping structure, not a simple pincer, but a complex arrangement of small spines that could seize and manipulate objects. Fossils show the proboscis could bend in multiple directions, suggesting sophisticated muscular control. But here's the problem. This structure doesn't correspond to any known arthropod appendage. Arthropods build their body parts by modifying leg segments. Antennae are modified legs. Mouthparts are modified legs. Even some reproductive structures are derived from leg segments. The proboscis doesn't follow this pattern. It seems to be something entirely new, a unique appendage that evolved specifically in Opabinia and then never appeared again. How Opabinia used this proboscis for feeding adds another layer of mystery. The mouth was located behind the proboscis, facing backward under the head. To eat, Opabinia would have needed to grab prey with the proboscis, curl it backward, and pass food into a rear-facing mouth. This feeding method is incredibly awkward compared to the forward-facing mouths of most predators. Why would such a convoluted system evolve? Some paleontologists suggest Opabinia fed while moving forward using the proboscis to pluck prey from the seafloor and deposit it directly into the mouth as it passed overhead. Others propose Opabinia was a scavenger, using its proboscis to probe sediments for buried organisms. The body structure of Opabinia defies standard arthropod classification. Its segmented body had lateral lobes extending from each segment, creating a fringed appearance. These lobes may have functioned as swimming paddles or respiratory surfaces, but they don't match the limb patterns of any known arthropod group. Most arthropods have jointed legs with specific numbers of segments and predictable arrangements. Opabinia's lobes were soft, unjointed, and arranged in overlapping rows. They're more reminiscent of annelid worm parapodia than arthropod limbs. Yet Opabinia was definitely not a worm. Classifying Opabinia has been a decades-long struggle. Initially, scientists tried to place it within known arthropod groups, perhaps a primitive crustacean or an early relative of trilobites. But detailed morphological studies kept revealing mismatches. Opabinia lacked the characteristic features of all major arthropod lineages. Its head didn't segment properly, its limbs didn't articulate correctly, and its eye arrangement was unprecedented. Some researchers proposed Opabinia represented a stem group arthropod, an early branch that split off before the major arthropod groups diversified. But this raised new questions. If Opabinia was ancestral to arthropods, why didn't any of its unique features appear in descendants? Modern phylogenetic analyses using both morphological and developmental data suggest Opabinia might belong to a completely extinct group of arthropod-like animals that explored alternative body plans during the Cambrian explosion. This group, sometimes called stem euarthropods or Cambrian weirdos, includes other bizarre creatures like Anomalocaris and Carigmacala. These animals share some features with arthropods: segmented bodies, hardened cuticles, complex eyes, but lack others and add unique characteristics found nowhere else. They represent evolutionary experiments that occurred during a brief window when body plans were still flexible and major groups hadn't yet locked into their modern forms. The preservation of Opabinia fossils is remarkable enough to reveal astonishing details. The Burgess Shale's exceptional conditions captured soft tissues, eye structures, and even gut contents. Analysis of these fossils shows Opabinia had a simple digestive tract running the length of its body, with no obvious specializations. Its gills, if it had them, left no clear trace. The internal anatomy is frustratingly simple, given the external complexity, offering few clues about how Opabinia's bizarre features functioned. One of the most intriguing recent discoveries involves Opabinia's developmental biology, or rather, our complete lack of understanding of it. Modern arthropods develop according to well-characterized genetic programs. Hox genes control segment identity, determining which segments produce which appendages. Eye development follows specific pathways involving PAX genes and other regulators. These genetic systems are highly conserved across arthropods. But how did Opabinia develop 5 eyes? How did it build a proboscis unlike any other appendage? The genetic toolkit required would need to include novel genes or novel uses of existing genes, innovations that apparently disappeared when Opabinia went extinct. Opabinia's ecology is equally mysterious. It lived in offshore marine environments with other Cambrian fauna, including predators like Anomalocaris and early arthropods. Fossil evidence suggests Opabinia was relatively common, not a rare oddity. This means its strange body plan was successful enough to maintain stable populations for millions of years. Yet nothing like it survived the Cambrian. As ecosystems stabilized and competition intensified, Opabinia's experimental design apparently couldn't compete with more conventional arthropod body plans. The extinction of Opabinia removed an entire branch of morphological possibilities from life's repertoire. The 5-eyed configuration, the flexible proboscis, the unique feeding method, all disappeared completely. No later animal ever reinvented these features or found ways to improve upon them. Opabinia represents an evolutionary path explored once and abandoned, a biological experiment preserved in stone, but impossible to replicate. The spiny paradox, Hallucigenia's upside-down decades. Few fossils have caused more confusion than Hallucigenia sparsa. This creature, roughly 2 centimeters long, was discovered in the Burgess Shale and initially seemed to represent an organism so bizarre that scientists literally couldn't figure out which end was up, which side was down, or even what kind of animal it was. For decades, Hallucigenia was reconstructed upside down and backward, walking on rigid spines while tentacle-like structures waved from its back. Only in the 1990s did researchers realize they'd had it completely wrong. The spines were defensive structures on top, and the tentacles were actually stubby walking legs underneath. But even with the orientation corrected, Hallucigenia remains deeply mysterious. But the biomechanics never quite worked. The spines were too rigid and poorly articulated to function as legs, and the tentacles showed no structures for capturing food. Something was wrong with this picture. The corrected orientation flipped everything. The organism walked on paired, clawed legs hidden along its underside, while the spines projected upward and outward as defensive structures. This made much more sense functionally. The legs had small claws for gripping surfaces, and the spines would deter predators trying to attack from above. But this solution created new mysteries. The legs weren't joined like arthropod limbs. They were soft, lobopod-style appendages, fleshy extensions with simple claws at the tips. These lobopods suggested Hallucigenia was related to modern velvet worms and tardigrades rather than to arthropods. Yet the spines were completely unlike anything those animals possess. The spines themselves are biomechanically puzzling. Each was a rigid, hollow structure tapering to a sharp point. They alternated in position along the body, creating a staggered defensive array. But how were they supported? Fossils show no obvious attachment points or muscles that could have controlled their orientation. They appear to have been fixed structures, like scales or external bone projections. Yet no modern organism builds spines this way. Hedgehogs, porcupines, and sea urchins all have spines rooted in skin or shell with muscular or hydraulic control systems. Hallucigenia's spines seem to be hollow extensions of the body wall, rigid but lightweight, projecting at fixed angles. Recent high-resolution imaging finally revealed Hallucigenia's head, and it was even stranger than expected. The anterior end featured a pair of simple eyes and a mouth opening ringed by tiny teeth. But behind the mouth was a circular structure that some researchers interpret as a pharynx studded with needle-like denticles. This throat armature doesn't match anything in modern animals. Pharyngeal teeth exist in some fish, and radula-like structures appear in mollusks. But Hallucigenia's throat spine seemed to be yet another unique innovation that evolved once and disappeared. The feeding behaviour of Hallucigenia remains unclear. The clawed legs suggest it walked along the seafloor, possibly climbing over microbial mats or small rocks. The position and structure of the mouth indicate it is fed by scraping or sucking food from surfaces. Some paleontologists propose it ate bacteria, algae, or organic detritus scraped from substrates. Others suggest it might have been a predator of even smaller organisms. But the throat spines complicate this picture. Were they for gripping food, processing it, preventing it from escaping? The biomechanics don't clearly support any single interpretation. Hallucigenia's relationship to other organisms represents one of paleontology's longest-running debates. It clearly belongs to the lobopodian group, animals with fleshy, unjointed legs, which includes ancestors of modern velvet worms. But Hallucigenia also shows features that might link it to early arthropods. The leg claws have structural similarities to arthropod claws, and the body segmentation hints at the repeated units seen in arthropods. Some researchers place Hallucigenia on the stem lineage leading to arthropods, suggesting it represents an intermediate form between soft-bodied lobopodians and hard-bodied, jointed arthropods. Others argue it's a specialized side branch that evolved unique features convergently. The spines create the biggest classificatory problem. No lobopodian, living or extinct, has structures like them. Velvet worms have tubercles and sensory papillae, but nothing resembling Hallucigenia's rigid spines. Tardigrades have claws and sometimes plate-like armour, but no elongated projections. The spines seem to be a unique invention, perhaps evolved specifically as a defence against Cambrian predators like Anomalocaris. This interpretation is supported by fossil evidence. Bite marks on other Cambrian organisms show that large predators were active and successful. In a world suddenly filled with hunters, defensive innovations would have been strongly selected. But here's the paradox. If the spines were such an effective defense, why didn't other organisms evolve similar structures? Why did this design disappear completely when Hallucigenia went extinct? Spines, scales, and armour became common later in evolution, appearing independently in countless lineages. Yet no animal ever recreated Hallucigenia's specific pattern. Hollow, rigid spines projecting from a soft body in staggered pairs. The design was apparently so specialised, so tied to Hallucigenia's specific ecology, that it couldn't be transferred or repurposed. The ecological context offers few clues. Hallucigenia lived in deep water environments alongside other strange lobopodians, early arthropods, and soft-bodied anomalies. Fossil assemblages show multiple individuals together, suggesting they weren't solitary. But were they social? Did they compete for resources? How did they interact with other species? The fossils provide snapshots of isolated moments. organisms frozen mid-stride, but reveal nothing about behaviour or ecology beyond basic morphology. Recent chemical analysis of hallucigenia fossils detected traces of organic compounds in the spines, suggesting they were covered by a thin cuticle similar to arthropod exoskeletons. This finding supports the hypothesis that hallucigenia was experimenting with the hardened body coverings that would later characterize arthropods. But it also raises questions. If the spines were the first steps toward an exoskeleton, why didn't the rest of the body follow suit? Why remain soft and vulnerable while developing isolated armored projections? The developmental biology of hallucigenia must have been extraordinarily complex. Building paired legs, alternating spines, segmented body units, and specialized head structures requires coordinated genetic programs. Modern arthropods use Hox genes and segmentation pathways to build their repeated body parts. Velvet worms use simpler, less rigidly controlled development. Hallucigenia somehow needed both systems: the precision of arthropod segmentation for its spines and the flexibility of lobopodian development for its soft body. How these systems coexisted in a single organism remains a complete mystery. Hallucigenia disappeared from the fossil record after the Cambrian, leaving behind only its bizarre fossilized forms and a legacy of confusion. For decades, it was the poster child for scientific misinterpretation, a cautionary tale about the difficulty of reconstructing extinct organisms. But even now, with better fossils and modern imaging techniques, hallucigenia keeps its secrets. It was neither arthropod nor worm, neither primitive nor advanced, but something uniquely itself. An evolutionary experiment that worked for a time, then vanished without teaching us why. The mineralization mystery, Cloudina's earliest shells. Before shells became common, before the great arms race between predators and prey armored the oceans, one organism made the first tentative step toward mineralized protection. Cloudina, living approximately 550 million years ago in the late Ediacaran period, constructed the oldest known skeletal structures in the animal kingdom. These weren't full shells, like later mollusks would build. They were small tubes, roughly the diameter of a pencil lead, made from stacked cone-shaped segments. But their very existence raises profound questions that paleontologists still cannot definitively answer. The tubes themselves are geometrically peculiar. Each Cloudina individual built a series of nested cones, like stacking paper cups inside each other. As the organism grew, it secreted calcium carbonate to form a new cone at the opening, creating a segmented tube that extended upward from the seafloor. The result looks somewhat like a miniature dunce cap or a wizard's hat made from stone. These tubes are preserved abundantly in late Ediacaran rocks from Namibia, China, and other locations, sometimes forming dense clusters that suggest colonial or gregarious behaviour. What makes Cloudina baffling is that we don't know what kind of animal built these tubes. The soft body that lived inside left no fossil trace. Only the mineralized shell remains. This is frustrating because the shell tells us Cloudina could secrete calcium carbonate, regulate crystallization, and construct complex three-dimensional structures. These capabilities imply sophisticated cellular machinery and genetic programs. But without soft tissue fossils, we can't determine if Cloudina was an early mollusk, a stem cornidarian, an ancient annelid, or something completely extinct. The chemical composition of Cloudina shells provides some clues, but raises new questions. The tubes are made from aragonite, a form of calcium carbonate also used by modern corals and many mollusks. But aragonite requires specific physiological conditions to crystallize. Organisms must maintain precise pH levels and calcium concentrations in the fluid where mineralization occurs. How did Cloudina, living before any modern animal groups had fully evolved, develop this capability? Did it evolve mineralization independently? Or was it inheriting genetic tools from even earlier, unknown ancestors. The arrangement of Cloudina shells in fossil deposits suggests the organisms were sessile, attached to one spot and unable to move. Some fossils show tubes growing from hard substrates like microbial mats, or even from other Cloudina tubes, creating small reef-like structures. This is significant because it represents some of the earliest evidence for animals creating three-dimensional habitats. But it also raises ecological questions. If Cloudina couldn't move, how did it feed? Most sessile marine animals today are filter feeders, using cilia or tentacles to capture drifting particles. Did Cloudina have feeding tentacles that extended from its tube? Were there cilia lining the opening? The fossils preserve no traces of soft feeding structures. One of the most intriguing aspects of Cloudina is evidence of predation. Some tubes show small, neat holes that appear to have been drilled or punched through the shell wall. These holes are remarkably consistent in size and shape, suggesting they weren't accidental damage, but deliberate attacks by predators. This would make Cloudina the victim of the oldest known predation in the fossil record. Direct evidence that the evolutionary arms race between attack and defense was already underway in the Ediacaran. But here's the mystery. What predator could drill through mineralized shells 550 million years ago? No known Ediacaran organism had hard mouthparts, drilling structures, or shell-crushing capabilities. The famous Cambrian predators like Anomalocaris wouldn't evolve for another 20 million years. Whatever attacked Cloudina must have had specialized feeding tools that left no fossil trace. Some researchers propose tiny, soft-bodied organisms with radula-like scraping structures. Others suggest acidic secretions that could dissolve calcium carbonate. Both explanations require complex adaptations that, like Cloudina itself, have no clear evolutionary context. The function of the shell is another unresolved question. Obviously, it provided protection. The drill holes proved that predators existed and targeted Cloudina. But the segmented, cone-in-cone construction seems overly complex for simple defense. Each segment slightly overlapped the previous one, creating a flexible but strong structure. As the tube grew, older segments near the base sometimes broke away, suggesting the shell wasn't permanent but constantly remodeled. Why build a shell this way? Why not create a simple, solid tube like modern tube worms? Some paleontologists propose the segmented design allowed Cloudina to adjust its height above the seafloor in response to sedimentation. If sediment buried the base, the organism could grow upward, keeping its feeding structures above the seabed. The cone-in-cone construction would make this easier than building a rigid tube. But this hypothesis assumes Cloudina needed to stay elevated, which circles back to the unknown feeding method. If it filtered particles from water, elevation would matter. If it absorbed nutrients directly like rangemorphs, height would be less important. The distribution of Cloudina fossils adds geographical puzzles. The organism appears in multiple continents, suggesting it could disperse widely across Ediacaran oceans. But how? Cloudina was sessile as an adult, unable to move. It must have had some larval or juvenile dispersal stage, tiny swimming or drifting forms that could travel in ocean currents before settling and building tubes. But no fossils of Cloudina larvae have ever been found. We don't know how they dispersed, how long they drifted, or how they chose settlement sites. The mineralization process itself represents a major evolutionary innovation. Building shells requires cells that can transport calcium and carbonate ions, control crystal growth, and lay down organic matrices that guide mineralization. These cellular systems are complex and require many specialized proteins. Modern shell-building animals use large suites of genes dedicated to biomineralization. Did Cloudina have similar genetic toolkits? If so, where did those genes come from? Did mineralization evolve multiple times independently? Or did Cloudina possess ancient biomineralization genes that were later co-opted by other lineages? The evolutionary relationships of Cloudina remain completely uncertain. Some researchers place it as a stem group Cnidarian based on its colonial growth and simple tube structure. Others argue for Anidarian affinities, pointing to potential similarities with modern tube-dwelling anemones. Still others suggest Cloudina might be a stem mollusk or an early member of an entirely extinct phylum. Phylogenetic analyses based solely on shell morphology cannot resolve these relationships because shells are such simple structures. They don't preserve enough anatomical detail for confident classification. Cloudina's fate is as mysterious as its origins. The organism disappeared at the end of the Ediacaran, just before the Cambrian explosion introduced a massive radiation of shell-bearing animals. Some species of tube-building organisms survived into the Cambrian, but none show clear evolutionary connections to Cloudina. The specific cone-in-cone architecture never reappeared. It's possible that Cloudina went extinct during the late Ediacaran environmental changes. It's equally possible that its descendants evolved different shell types and are hidden among Cambrian fossils we've already found, but haven't properly identified. What we're left with is a pioneering organism that invented shells, faced the earliest predators, built reef-like communities, and dispersed across oceans, all while remaining anatomically unknown. Cloudina proves that biomineralization, colonial living, and predator-prey dynamics were already sophisticated 550 million years ago. But it raises more questions than it answers, standing as a mineralized monument to how much we still don't understand about early animal evolution. The locomotion riddle, Kimberella's sliding tracks. Among Ediacaran fossils, Kimberella quadrata stands out for one remarkable feature. It left tracks. While most Ediacaran organisms were sessile or slow-moving at best, Kimberella created clear trace fossils showing it could move across the seafloor in what appears to be directed, purposeful motion. These tracks, preserved alongside body fossils in Russia and Australia, suggest Kimberella was more advanced than its contemporaries. But the mechanics of how it moved, what it was doing, and what kind of organism it was, remain frustratingly unclear despite decades of study. Kimberella fossils appear as oval impressions, typically 5 to 15 centimeters long, with a quilted or striated surface texture. One end shows a broader, more structured appearance that researchers interpret as the front, while the opposite end tapers slightly. This front-back asymmetry is significant because it indicates bilateral symmetry. Kimberella had distinct anterior and posterior ends, unlike the radial symmetry of jellyfish or the ambiguous geometry of other Ediacaran organisms. Bilateral symmetry is associated with directed movement and active lifestyles, suggesting Kimberella was more mobile than most of its neighbours. The trace fossils associated with Kimberella are what make it truly intriguing. Preserved near body fossils are scratch marks and grooves arranged in patterns that suggest scraping or grazing behaviour. These marks extend in front of the body impressions, indicating Kimberella moved forward while feeding, creating a characteristic feeding trace. Some researchers interpret these patterns as evidence for a radula, the rasping, tooth-covered tongue structure used by mollusks to scrape algae and organic material from surfaces. If true, this would make Kimberella the earliest known mollusk or mollusk relative, pushing the origins of this hugely successful group back into the Ediacaran. But here's the problem. We don't actually know if Kimberella had a radula. The scratches could be produced by other structures, stiff bristles, hardened edges, or muscular ridges on the underside of the body. No Kimberella fossil has preserved the actual feeding apparatus, only the marks it left behind. The interpretation of these traces as radulae marks is based on comparison with modern mollusk feeding traces. But Kimberella lived in a fundamentally different world, with different ecological rules. Assuming it fed like a modern mollusk, maybe projecting our understanding backward onto an organism that worked in completely different ways. The body structure of Kimberella adds to the uncertainty. The quilted surface texture seen in fossils might represent external ridges, internal muscle bands, or simply the way soft tissues compressed into sediment. Some researchers see evidence for a tough, protective outer layer similar to mollusk mantles. Others interpret the same features as simple folds in a worm-like body. Three-dimensional reconstructions based on fossils offer competing models. Some show Kimberella as a slug-like crawler with a muscular foot, others as a flat, gliding organism more similar to certain flatworms. The question of locomotion is central to understanding Kimberella. How did it move? Modern mollusks like slugs and snails use a muscular foot that produces waves of contraction, allowing them to glide smoothly over surfaces. This requires sophisticated neuromuscular coordination and secretion of mucus to reduce friction. Did Kimberella have these capabilities? The trace fossils show it could move in relatively straight lines and make turns, suggesting some level of directional control, but the speed and mechanism remain unknown. The traces don't show clear signs of foot waves or the continuous trail of mucus-like secretions seen in modern gastropod tracks. Another possibility is that Kimberella moved through ciliary gliding using microscopic hair-like cilia covering its underside to propel itself forward. This method is used by many flatworms and requires far less complex musculature than foot-based locomotion. Ciliary gliding can produce smooth, directed movement and would leave behind the kinds of tracks associated with Kimberella. But there's no way to tell from fossils whether cilia were present. Cilia are microscopic and made of soft proteins. They don't fossilize under normal conditions. The feeding ecology of Kimberella is equally mysterious. The scrape marks suggest it was eating microbial mats, the thick bacterial carpets that covered Ediacaran seafloors. These mats were nutritious and abundant, making them an obvious food source. But how did Kimberella process this food? Did it have a gut for digestion? Modern bilaterians have through guts with a mouth at one end and an anus at the other, allowing continuous feeding and efficient nutrient extraction. Some Kimberella fossils show possible evidence for internal structures that might be gut traces, but these are highly ambiguous. Other researchers see no clear signs of internal anatomy at all. The evolutionary position of Kimberella has been hotly debated. If it was a stem mollusk, it represents an incredibly early stage in mollusk evolution, before shells, before true raduli, possibly before many defining mollusk features evolved. This would push the mollusk lineage much deeper into the past than traditional phylogenies suggested. But other analyses place Kimberella outside the mollusk lineage entirely, perhaps as a stem bilaterian, an early bilateral animal that predated the split between mollusks, annelids, arthropods, and other groups. This interpretation makes Kimberella even more important because it would be one of the earliest mobile bilaterians, representing a crucial transition in animal evolution. Recent chemical analyses of Kimberella fossils have detected organic compounds, but nothing specific enough to definitively assign it to a particular group. The preservation is simply too poor to extract clear molecular signatures. This means classification must rely on morphology alone, the shape of the body, the pattern of traces, the texture of surfaces. But morphology can be misleading, especially in soft-bodied organisms that fossilize poorly. The disappearance of Kimberella at the end of the Ediacaran adds another layer of mystery. If it was an early mollusk, why didn't it survive into the Cambrian when mollusks rapidly diversified? If it was a stem bilaterian, what happened to its lineage? The Cambrian explosion produced a wealth of new bilaterians with shells, complex organs, and sophisticated behaviors. Yet nothing in the Cambrian clearly descends from Kimberella. Its grazing lifestyle and bilateral body should have been advantageous, but it vanished anyway. One possibility is that Kimberella was outcompeted by Cambrian animals with better adaptations, harder shells, more efficient feeding structures, or improved mobility. Another is that environmental changes made microbial mats less common, removing Kimberella's food source. Cambrian burrowers disrupted seafloor mats, and new grazing animals may have consumed them faster than they could regenerate. Whatever the cause, Kimberella's extinction removed one of the most promising early experiments in bilateral animal design. What remains is a tantalizing glimpse of mobility and feeding behavior emerging in ancient seas. Kimberella shows us that complex directed movement existed before the Cambrian explosion, that bilateral animals were already exploring different lifestyles, and that feeding specializations were evolving before mineralized skeletons appeared. But it refuses to reveal its true nature. Was it a mollusk ancestor? A stem bilaterian? Something completely extinct? The tracks tell us it was there, moving and eating, but they can't tell us what it really was. The skeleton enigma, Chancelloria's crystalline armor. Chancelloria is a Cambrian fossil that looks like it was designed by a committee that couldn't agree on what kind of animal to make. Its body was roughly oval or cucumber-shaped, covered entirely in star-shaped skeletal elements called sclerites. Each sclerite was a tiny, intricate structure made of calcium carbonate, radiating multiple rays from a central point like miniature caltrops or jacks used in children's games. Thousands of these sclerites interlocked across Chancelloria's surface, creating a complete coat of crystalline armor. But here's the puzzle. We have no idea what kind of animal was underneath that armor, how it lived, or where it fits in the tree of life. The sclerites themselves are marvels of biological engineering. Each one consists of multiple rays meeting at a central node, with the rays extending outward and sometimes branching. The rays weren't solid. They were hollow tubes with walls made of carefully organized calcium carbonate crystals. This construction created lightweight but strong defensive structures. The pattern and number of rays varied. Some sclerites had 3 rays, others 7 or more. The diversity suggests different sclerite types served different functions or covered different body regions. But we can't correlate them with specific anatomical features because the soft body never fossilized. Finding complete Chancelloria specimens is rare. Usually, paleontologists encounter isolated sclerites scattered through Cambrian rocks. These tiny skeletal elements are durable and fossilize well, but they've become disconnected from the animals that produce them. It took years of careful excavation and lucky discoveries to find articulated specimens showing how sclerites were arranged on the living animal. The articulated fossils revealed that sclerites covered the entire body surface in overlapping layers, creating what must have been formidable protection against Cambrian predators. But protection from what? Chancelloria lived during the Cambrian when the first major predators were evolving. Anomalocaris and other radiodonts were hunting with grasping appendages and complex jaws. Chancelloria's armor would have made it difficult to bite or grab. The sharp rays projecting in all directions would deter most attacks, working like a miniature hedgehog's quills. Yet some Chancellorian fossils show signs of damage or partial sclerite loss, suggesting predators did successfully attack them. What could bite through or pull apart crystalline armor? We don't know, but something did. The soft body anatomy of Chancelloria is almost entirely unknown. The sclerite-covered exterior preserved well, but the internal organs, tissues and structures left no trace. We don't know if Chancelloria had a mouth, gut, circulatory system or nervous system. We can't tell if it was mobile or sessile. Some researchers propose it was a sponge-like organism, anchored to the seafloor and filter feeding by pumping water through its body. The sclerites would have protected the delicate filtering tissues. But other scientists point out that the sclerite arrangement doesn't leave obvious openings for water flow, making the sponge hypothesis problematic. Another theory suggests Chancelloria was a stem group deuterostome, an early relative of modern echinoderms and chordates. This classification is based on very subtle features of sclerite microstructure and the way they're arranged. But this hypothesis is controversial because it requires Chancellorhea to be related to animals that later evolved radically different body plans. How would a sclerite armoured cucumber-shaped organism give rise to starfish or sea urchins? The evolutionary pathway isn't clear. A third possibility is that Chancellorhea represents an entirely extinct phylum, a major branch of the animal tree that evolved, thrived for a while, and then disappeared completely. This would make Chancellorhea's sclerites a unique evolutionary invention, tried once and never repeated. Sclerite-based armour appeared in several other Cambrian animals, but none quite like Chancellorhea's star-shaped designs. Each species seemed to be experimenting with different approaches to skeletal protection, and most of these experiments didn't last beyond the Cambrian. The mineral composition of Chancellorhea sclerites adds to the mystery. They're made of calcium carbonate, specifically calcite crystals arranged in precise orientations. The crystals grow in organised patterns that must have been biologically controlled. The organism had to regulate crystal nucleation, growth rates, and spatial arrangement. This requires sophisticated cellular machinery, including specialised proteins and controlled chemical environments. How Chancellorhea developed this capability is unknown. Did it evolve biomineralization independently, or inherit ancient genetic tools from even earlier ancestors? The diversity within the Chancellorhea genus raises more questions. Different species had different sclerite designs, some with more rays, others with branching patterns, still others with unusual shapes. This diversity suggests Chancellorhea was adapting to different environments or ecological niches. But what were those niches? All Chancellorhea fossils come from marine environments, mostly from deposits representing offshore or deeper waters. Did different species live at different depths? Were they specialised for particular current regimes or substrate types? Without soft tissue preservation, we can't test these hypotheses. Recent discoveries of Chancellorhea-like organisms from other Cambrian fossil deposits have expanded the picture but not clarified it. These relatives show similar sclerite-based body coverings, but with variations in size, shape, and sclerite design. Some were larger than Chancellorhea, others much smaller. Some had sclerites arranged in obvious geometric patterns, others in seemingly random arrangements. This diversity suggests the sclerite armour strategy was successful and led to multiple species, but then all of them disappeared. The extinction of Chancellorhea and its relatives occurred during the Middle to Late Cambrian, a period of major ecological restructuring. Predators were becoming more sophisticated, developing harder jaws and more powerful bite forces. Prey species were evolving better defences, thicker shells, better mobility, burrowing behaviours. Chancellorhea's sclerite armour, impressive as it was, may not have kept pace. If predators evolved tools to break or pry apart sclerites, the entire defensive strategy would fail. Another possibility is that Chancellorhea lost out in competition for space or resources. The Cambrian seafloor was becoming crowded with diverse organisms, many competing for similar ecological roles. If Chancellorhea was sessile and relied on filter feeding, it would compete with sponges, early bryozoans, and other suspension feeders. If it was mobile, it would compete with early mollusks, arthropods, and worms. With limited advantages beyond its armour, Chancellorhea may have simply been outcompeted. What makes Chancellorhea especially frustrating to paleontologists is how much information its fossils contain, and how little we can extract. The sclerites preserve beautifully, down to microscopic details. We can measure crystal structures, growth patterns, and chemical compositions. We can count sclerites, map their arrangements, and reconstruct the external appearance with confidence. But all of this tells us almost nothing about what Chancellorhea actually was. Without soft tissues, without developmental stages, without living relatives for comparison, we're left staring at crystalline armour, with no way to see what wore it. The segmentation problem, Spriggina's head-tail ambiguity. Spriggina floundaris looks like it should be simple to understand. Its fossils show a clear body plan, a crescentic head shield followed by dozens of repeated segments extending backward in a tapering series. The whole organism, typically 5 to 10 centimetres long, resembles a trilobite, or perhaps an early arthropod. Many paleontologists initially classified it as an arthropod relative, interpreting the head structure as a primitive cephalon and the segments as early body modules. But decades of study have revealed fundamental problems with this interpretation, and Spriggina has become one of the most debated Ediacaran fossils. The head region is the source of much confusion. It's crescentic or horseshoe-shaped, wider than the body segments and distinctly different in texture. Some researchers see this as a head shield with sensory structures similar to trilobite cephalons. Others interpret it as a feeding apparatus, perhaps a hood that could expand and contract to capture food. Still others suggest it might not be a head at all, but a specialized anterior structure used for anchorage or propulsion. The fossil preservation doesn't resolve these alternatives. The head appears as a smooth impression with no internal details. The body segments present their own mysteries. They're arranged in 2 columns running down the organism's length, offset from the midline in what's called isomeric segmentation. This pattern is unusual. Most segmented animals have segments that meet at the midline or show clear left-right pairing. Spriggina's segments are staggered, creating a zigzag effect along the body's center line. This asymmetric arrangement doesn't match arthropod segmentation, annelid segmentation, or any pattern seen in living animals. Some paleontologists argue the staggered segments are an artifact of preservation. Perhaps the organism was twisted or compressed in a way that made symmetric segments appear offset. But this explanation has problems. Multiple Spriggina fossils from different locations show the same staggered pattern. If it were just preservation or distortion, we'd expect to see both symmetric and asymmetric specimens, depending on how each individual was buried. The consistency suggests the offset pattern was real. The function of these segments is equally unclear. In modern arthropods, body segments can bear limbs, house organs, or provide attachment points for muscles. Did Spriggina's segments serve any of these functions? Fossils show no evidence of appendages, though that doesn't necessarily mean they didn't exist. Soft limbs might not fossilize. Some researchers propose the segments were muscle blocks that produced undulating movements, allowing Spriggina to swim or crawl. Others suggest they were primarily structural, providing flexibility while maintaining body integrity. The tail region tapers to a point, with segments becoming progressively smaller toward the posterior end. This tapering is consistent with directed locomotion. Organisms with distinct heads and tails typically move head first, but determining which end was actually the front requires knowing where the mouth was located, and no Spriggina fossil has preserved a mouth opening. The crescentic structure is assumed to be anterior based on its complexity and the tapering posterior, but this is an assumption, not confirmed fact. Trace fossils that might be associated with Spriggina would be incredibly valuable for understanding how it moved, but no definitive traces have been linked to it. The Ediacaran seafloor was covered in microbial mats, and many organisms left feeding traces or movement tracks. Some traces in deposits where Spriggina occurs might have been made by it, but without the organism preserved at the end of a trace, we can't be certain. This lack of behavioural evidence leaves us guessing about lifestyle and ecology. The evolutionary position of Spriggina has ping-ponged between different hypotheses for decades. Early interpretations placed it as a stem arthropod, an early member of the lineage that would later include trilobites, insects, and crustaceans. This made sense given the segmented body and apparent head shield, But closer examination revealed too many differences. The segment arrangement was wrong, the head structure didn't match, and there were no signs of jointed limbs or other arthropod characteristics. Alternative hypotheses suggested Sprigina might be an early annelid, segmented worm, or a stem group bilaterian that predated the split between major animal lineages. These classifications are based more on Sprigina's segmentation than on positive evidence linking it to any group. Segmentation is a powerful developmental feature that evolved multiple times in different lineages, so shared segmentation doesn't necessarily indicate a close relationship. More radical proposals place Sprigina outside the animal kingdom entirely, suggesting it was a colonial organism made of individual units stacked together, or perhaps a complex protozoan that evolved multicellularity independently. These ideas are speculative and have little supporting evidence, but they highlight how difficult Sprigina is to classify. When an organism can be plausibly argued to be an arthropod, a worm, or something not quite animal, it reveals the limits of our classification systems. Recent morphometric studies using three-dimensional scanning and statistical analysis of multiple specimens have revealed subtle details previously overlooked. The segments show slight variations in width, spacing, and surface texture along the body length. Some segments appear to have small ridges or furrows that might represent attachment points for internal structures. But these observations don't clarify function. They just show that Sprigina had complex anatomy hidden beneath the simple surface appearance. The size range of Sprigina fossils suggests genetic growth. Individuals started small and grew larger over time. But we don't have clear developmental series showing how this growth occurred. Did Sprigina add new segments as it grew, like many arthropods and annelids do? Or did existing segments simply enlarge? If it added segments, where were they added? At the posterior end, anterior end, or distributed throughout the body? These questions can't be answered without preserving growth stages or individuals caught mid-molt or mid-transformation. The ecological role of Sprigina is another mystery. Was it a predator, a scavenger, a filter feeder, or something else? The head structure might have housed a mouth, but we don't know what kind. A predatory lifestyle would require some method of capturing and handling prey, grasping appendages, a toothed pharynx, or other tools. There's no evidence for any of these. A filter-feeding lifestyle would require systems to move water and capture particles, which also aren't visible. Sprigina could have been a deposit feeder, consuming sediment and extracting nutrients. But this is speculation based on analogy with modern organisms, not on direct fossil evidence. The disappearance of Sprigina at the end of the Ediacaran adds to its enigmatic nature. It wasn't simply replaced by something better adapted, it vanished entirely, along with most other Ediacaran fauna. The Cambrian explosion introduced many segmented animals, but none clearly descended from Sprigina. Its particular combination of features, the crescentic head, the staggered segments, the tapering body, never reappeared in later evolution. If Sprigina was indeed an early step toward arthropods or annelids, it was a step down a path that dead-ended, leaving no trace in descendants. What we're left with is an organism that looks tantalizingly familiar, yet remains fundamentally mysterious. Sprigina has segments like many successful animals, a head-like structure suggesting complexity, and a body plan that implies directed movement. But it won't fit into any category we understand. It's a reminder that the early history of animal evolution included experiments that don't map onto modern groups, body plans that were tried and abandoned, and organisms that remain biological riddles despite being preserved in exquisite detail. The lifestyle puzzle, Charnia's deep-water existence. Charnia masoni presents a paradox that has puzzled paleontologists since its discovery in the 1950s. This frond-like organism, one of the earliest complex life forms known from the Ediacaran period, lived in deep, dark waters where photosynthesis was impossible. Yet it grew to impressive sizes, sometimes exceeding a metre in length, with intricate branching structures that superficially resembled ferns or sea pens. The fundamental question remains unanswered. How did Chania obtain energy in an environment with no light and limited food sources? Chania's body plan was constructed from repeated branching units arranged along a central axis. Each side branch split into smaller branches, which split again, creating a fractal-like pattern that maximized surface area. This design is identical to rangeomorphs described earlier, placing Chania firmly within that mysterious group. The entire organism was anchored to the seafloor by a disc-shaped holdfast, keeping it stationary throughout its life. The frond extended upward into the water column, exposing its branching surfaces to whatever nutrients or energy sources were available. The deep water setting is confirmed by sedimentological analysis of rocks containing Chania fossils. These rocks formed in environments below the photic zone, the depth where sufficient light penetrates for photosynthesis. Modern deep-sea environments support life through chemosynthesis, bacteria that derive energy from chemicals like hydrogen sulfide or methane. Some researchers propose Chania hosted chemosynthetic bacteria in its tissues, forming a symbiotic relationship similar to modern tube worms at hydrothermal vents. The bacteria would provide energy, and Chania would provide a structure and surface for bacterial growth. But this hypothesis has problems. Chemosynthesis requires specific chemical conditions, concentrated sources of reduced compounds that bacteria can oxidize. Hydrothermal vents provide these conditions, but they're localized and rare. Chania fossils occur in widespread deposits representing normal deep-sea conditions, not vent environments. Where would the chemical energy come from? Some researchers suggest methane seeps or sulfide-rich sediments could have provided localized energy sources, but evidence for such conditions in Chania deposits is limited. Another possibility is osmotrophy, directly absorbing dissolved organic molecules from seawater. The Ediacaran oceans may have contained higher concentrations of dissolved organics than modern seas, products of bacterial activity and decaying organic matter. Chania's high surface area structure would be ideal for absorbing these nutrients. But as discussed with rangeomorphs, scaling this feeding method to metre-sized organisms poses biophysical challenges. Diffusion rates and nutrient concentrations would need to be far higher than what seems realistic. The ecological setting of Chania adds another dimension to the mystery. Fossil assemblages show Chania lived in dense aggregations, with hundreds of individuals clustered together in relatively small areas. This distribution is strange for an organism relying on dissolved nutrients. Crowding should create competition, with individuals depleting resources around them and limiting each other's growth. Yet Chania clearly thrived in these dense populations. Either nutrients were so abundant that competition didn't matter, or Chania obtained energy through some unknown mechanism that wasn't resource limited in the usual sense. Recent studies have examined the orientation of Chania fossils relative to ancient currents. The fronds consistently align with flow directions, suggesting they position themselves to maximize exposure to moving water. This alignment would make sense for filter feeding or nutrient absorption. Currents bring fresh water with dissolved resources, but it could also support chemosymbiosis if bacteria-rich water flowed from nearby chemical sources. Unfortunately, fossil orientation alone can't distinguish between these possibilities. The growth patterns preserved in Chania fossils offer tantalizing hints. Some specimens show distinct banding or rings in their fronds, similar to tree rings. These bands might represent growth increments, suggesting Chania grew periodically rather than continuously. If so, what triggered growth episodes? Seasonal changes in deep water are minimal. No light cycles, limited temperature variation. Perhaps growth responded to episodic food availability, nutrient pulses from overlying surface waters, or biological rhythms independent of environmental cues. Size variations among Chania fossils also raise questions. Individuals range from a few centimetres to over a metre, with no obvious size classes or discrete life stages. This suggests continuous growth and variable lifespans. But how long did Charnia live? Modern deep-sea organisms often grow slowly and live for decades or even centuries. Was Charnia similarly long-lived? Did it reproduce sexually, vegetatively, or both? No Charnia fossils show clear reproductive structures or evidence of budding offspring. The holdfast structure provides another clue to Charnia's biology. These disc-shaped attachment structures were apparently organic, not mineralized, and they firmly anchored the organism to substrates that range from seafloor mud to hard microbial mats. The strength of attachment must have been considerable. Deep water currents could be strong, and a metre-tall frond would catch substantial water flow. Yet Charnia apparently remained anchored throughout its life, suggesting robust attachment systems. One possibility rarely discussed is that Charnia was dead for most of its existence. Perhaps it only lived briefly as a small, active organism that built the frond structure, then died while the structure remained standing. This would be analogous to corals, where living tissue forms a calcium skeleton that persists long after the organism dies. But Charnia wasn't mineralized, it was entirely soft-bodied. Could soft structures remain intact for extended periods in deep water? Modern observations suggest bacterial decomposition would quickly break down unprotected organic material unless preserved by specific chemical conditions. The evolutionary relationships of Charnia remain as mysterious as its ecology. As a rangemorph, it shares characteristics with other fractal-bodied ediacaran organisms, but this grouping is based more on shared morphology than on clear phylogenetic evidence. Genetic relationships are impossible to determine without DNA or clear anatomical homologies. Charnia could be an early animal experimenting with colonial living, a giant protozoan that evolved complex structures, or something entirely outside our modern classification schemes. The extinction of Charnia and its relatives adds to the mystery. These organisms disappeared completely at the end of the Ediacaran, leaving no obvious descendants. The Cambrian explosion introduced new ecological dynamics: mobile predators, burrowing animals, bioturbation that disrupted seafloor communities. Perhaps these changes destroyed the stable, low-energy ecosystems where Charnia thrived. Or perhaps environmental shifts eliminated whatever chemical or nutrient sources Charnia depended on. What remains is a beautiful, complex fossil organism that doesn't fit any ecological or evolutionary category we understand. Charnia lived where nothing should have grown so large, obtained energy through unknown mechanisms, reproduced by mystery methods, and vanished without leaving instructions for how it worked. It stands in museum collections and fossil beds as a permanent reminder that Earth's history includes forms of life whose basic biology remains beyond our comprehension. The eye evolution paradox, Anomalocaris' compound vision. When paleontologists first discovered complete Anomalocaris specimens in the 1980s, they recognized a predator built for Cambrian dominance: powerful grasping appendages, flexible body, and large size. But discoveries in the 2000s revealed something unexpected. Anomalocaris had the most sophisticated eyes of any Cambrian organism, with up to 16,000 individual lenses arranged in compound structures rivaling those of modern dragonflies. This raises a profound question. How did such advanced eyes evolve so early in animal history? And why doesn't the fossil record preserve the intermediates leading up to them? The eyes of Anomalocaris were mounted on stalks extending from the sides of the head, giving them a wide field of view and possibly stereoscopic vision. Each eye contained thousands of individual ommatidia, optical units consisting of lenses and light-detecting cells. Fossilized eyes from specimens found in Australia preserve the lens arrangements with astonishing clarity. Scientists can count individual lenses and map their organization, revealing sophisticated optical designs that shouldn't exist in animals from 520 million years ago. Modern compound eyes are products of complex developmental programs involving many genes and coordinated tissue differentiation. PAX genes control eye formation, while other genetic networks determine lens arrangement, photoreceptor types, and neural wiring. These systems are highly conserved across arthropods, suggesting they evolved early and remain stable. But Anomalocaris predates most arthropod groups, living before the genetic toolkit for compound eyes should have been fully developed. Either Anomalocaris represents a surprisingly late stage in eye evolution, with earlier forms undiscovered, or eyes evolved faster and earlier than current models suggest. The optical quality of Anomalocaris eyes approaches modern standards. Analysis of lens geometry shows they could have produced relatively sharp images, useful for detecting prey, avoiding predators, or navigating complex environments. The spacing and curvature of lenses suggests sensitivity to movement and possibly color discrimination. This level of sophistication implies significant brain investment. Processing input from 16,000 lenses requires neural networks capable of image integration, pattern recognition, and behavioral responses. Did Anomalocaris have brain structures comparable to modern predatory arthropods? Preserved fossils show that Anomalocaris brains were relatively large for a Cambrian animal, with distinct lobes possibly dedicated to visual processing. But soft neural tissue doesn't preserve the structural details needed to understand function. We can measure brain size and overall shape, but we can't trace neural connections or identify specific processing centers. The cognitive capabilities of Anomalocaris, how it perceived its world, how quickly it could respond to visual stimuli, whether it had any capacity for learning or memory, remain entirely unknown. The existence of such advanced eyes in Anomalocaris implies that prey animals must have already evolved defenses requiring sophisticated vision to overcome. Complex eyes only evolve when there's strong selective pressure for improved vision, typically in predator-prey arms races. This means Cambrian ecosystems were already experiencing intense visual predation pressures, with prey evolving camouflage, armor, or speed to escape, and predators evolving better eyes to catch them. But when did this arms race start? The earliest Cambrian fossils already show diverse eye types, suggesting vision evolution occurred rapidly before the period began. The diversity of eye types in the Cambrian is itself puzzling. Anomalocaris had compound eyes, but other animals evolved completely different designs. The camera-like eyes of some mollusks, the simple pit eyes of worms, the bizarre tower eyes of some trilobites. This diversity suggests multiple independent origins for vision or extremely rapid divergence from a common ancestral eye. Either scenario requires eye evolution to be much faster and more flexible than observed in later periods. Why was the Cambrian such a hotbed of visual innovation? One hypothesis proposes that environmental changes triggered eye evolution. Rising oxygen levels in Cambrian oceans may have cleared the water, increasing visibility and making vision suddenly useful. Alternatively, the evolution of hard body parts created new visual targets, shiny shells, colorful markings, or distinctive shapes that could be seen from a distance. These environmental and ecological changes could have created conditions where vision offered unprecedented advantages, driving rapid evolution of increasingly sophisticated eyes. But this still doesn't explain the absence of intermediate forms. Between simple, light-sensing patches and Anomalocaris' 16,000 lens, compound eyes lie many potential steps: clusters of photocells, primitive lenses, partially organized ommatidia. Yet very few fossils preserve these transitional stages. The earliest eyes in the fossil record are already quite sophisticated. Either the transitional stages occurred so rapidly they left few fossils, or they occurred in organisms with soft bodies that didn't fossilize, or there's something fundamentally wrong with our understanding of how eyes evolved. Recent discoveries of fossilized eyes from other Cambrian animals have begun filling some gaps. Simple eyes with just a few dozen lenses have been found, along with intermediate forms showing various organizational schemes. These findings suggest eye evolution was more gradual than Anomalocaris alone suggests, but significant gaps remain. The genetic changes required to go from simple to complex eyes involve changes in developmental timing, spatial patterning, and cell differentiation. Modelling these evolutionary transitions is difficult without knowing the ancestral conditions or having living intermediates to study. The preservation of Anomalocaris eyes themselves is remarkable and puzzling. Eyes are delicate structures, lenses are thin, photoreceptor cells are fragile, and the whole apparatus degrades rapidly after death. That any Cambrian eyes fossilized at all requires exceptional circumstances. The Emu Bay Shale in Australia, where the best preserved Anomalocaris eyes were found, represents unusual conditions, where rapid burial and specific water chemistry prevented decay and allowed soft tissues to mineralize. Without these rare preservational windows, we'd have no idea about Cambrian eye evolution at all. This raises an uncomfortable question. How much are we missing? If Anomalocaris eyes only fossilized under exceptional conditions, what percentage of Cambrian eye diversity went unpreserved? There could have been dozens of other eye types, earlier evolutionary stages, or extinct visual systems that left no trace. Our understanding of early vision evolution is based on a tiny sample of organisms that happen to die in the right places under the right conditions. The true story could be far more complex than fossils allow us to see. The cognitive implications of advanced vision also deserve consideration. Eyes are expensive to build and maintain. They require continuous protein synthesis, energy for photoreceptor operation, and brain tissue for image processing. Anomalocaris wouldn't have evolved such costly eyes unless it had behaviours that justified the investment. This implies complex hunting strategies, perhaps ambush predation or pursuit hunting that required tracking prey movements. Did Anomalocaris exhibit learning? Could it remember successful hunting locations or recognise individual prey types? These cognitive questions can't be answered from fossils alone. The extinction of Anomalocaris and its relatives removed these early sophisticated eyes from the evolutionary stage. While compound eyes persisted and diversified in later arthropods, the specific design Anomalocaris used disappeared. Modern arthropods reinvented compound eyes using presumably similar genetic toolkits, but whether they inherited any of Anomalocaris' specific adaptations remains unclear. The radiodonts represent an independent experiment in visual predation that succeeded brilliantly for a time, then ended without passing its innovations forward. The reproductive riddle, Waiwaxia's unknown life cycle. Waiwaxia corrugata looked like an armoured slug crawling out of a fever dream. Its back was covered in overlapping scales and prominent spines. Its underside was soft and presumably muscular, and its feeding apparatus consisted of hooked structures that might have been an early radula. Numerous fossils exist, many beautifully preserved in the Burgess Shale, showing intricate details of armour arrangement and scale microstructure. But despite this wealth of specimens, we know absolutely nothing about how Waiwaxia reproduced. No eggs, no larvae, no juveniles, no reproductive structures whatsoever. The size range of Waiwaxia fossils spans from small individuals a few centimetres long to larger specimens exceeding 10 centimetres. This range suggests growth over time, but we can't distinguish between juveniles and adults. There are no distinct size classes, no obvious morphological differences that might separate immature from mature individuals. Either Waiwaxia reproduced at multiple sizes throughout life, or the reproductive features were entirely soft and left no fossil traces. Modern mollusks, which Waiwaxia might be related to, show incredible diversity in reproductive strategies. Some release eggs and sperm into the water for external fertilization. Others brood embryos internally and give birth to miniature adults. Still others produce specialized larval stages that look nothing like adults, swimming, feeding larvae that eventually metamorphose. Any of these strategies could have been used by Waiwaxia, but without preserved reproductive structures or larvae. We're guessing. The armor plates, called sclerites, show growth patterns that might offer clues. Individual sclerites appear to have grown incrementally, adding material at their edges. Some show damage and repair, suggesting the animal could survive injuries and regenerate damaged scales. But sclerite growth doesn't directly inform reproduction. Did Wiwaxia add new sclerites as it grew? Or did existing sclerites simply enlarge? If it added new sclerites, where did they appear? Along the body margin interspersed between existing scales, or in specific growth zones? The absence of juvenile fossils is particularly puzzling. Burgess Shale deposits contain thousands of beautifully preserved organisms, many represented by multiple life stages. Yet small Wiwaxia fossils don't look obviously juvenile. They appear as miniature adults with fully formed armor and spines. Either Wiwaxia juveniles were morphologically similar to adults from very early in development, or they lived in different habitats where they didn't fossilize, or they had soft-bodied larval stages that left no traces. If Wiwaxia had a larval stage, what would it have looked like? Modern mollusk larvae are typically microscopic, or small enough that fossilization would be extremely rare. They swim in the water column, feed on plankton, and eventually settle to the seafloor, where they metamorphose into adult forms. A similar life cycle in Wiwaxia would explain the absence of juvenile fossils, but it would raise new questions. How would a soft larva transform into a spiny, armored adult? This metamorphosis would require extensive tissue reorganization and sclerite formation, complex processes that require sophisticated developmental control. The reproductive ecology would also affect population dynamics. If Wiwaxia broadcast spawned like many marine invertebrates, producing thousands of tiny eggs released into the water, most offspring would die before reaching adulthood. This is a high-waste but effective strategy for colonizing new areas and maintaining populations. Alternatively, if Wiwaxia produced fewer offspring but invested more in each one, brooding eggs or providing parental care, survival rates would be higher, but dispersal more limited. Different strategies have different ecological implications, and we can't determine which Wiwaxia used. The geographic distribution of Wiwaxia fossils might offer indirect evidence. The organism appears in deposits representing similar environments across limited geographic areas, suggesting it couldn't disperse widely. This might argue against larval stages capable of long-distance drift, but absence of evidence isn't evidence of absence. Wiwaxia might have had wide dispersal, but simply didn't colonize other fossilized environments, or fossils from other locations haven't been found yet. Another consideration is seasonality. Many modern marine invertebrates reproduce seasonally, timing spawning to coincide with favorable conditions. Did Wiwaxia reproduce year-round, or only during specific seasons? The Cambrian oceans probably had seasonal variations in temperature, nutrient availability, or current patterns. If Wiwaxia timed reproduction to environmental cycles, this would imply sensory systems capable of detecting seasonal cues. But without living specimens or detailed ecological data, this is pure speculation. Sexual versus asexual reproduction presents another unknown. Did Wiwaxia have separate sexes, or were individuals hermaphroditic? Modern mollusks exhibit both systems. Some are strictly male or female, others are simultaneous hermaphrodites, both male and female organs at once, and still others are sequential hermaphrodites, changing sex during life. Each system has advantages and costs. Separate sexes require finding mates, but avoid self-fertilization problems. Hermaphroditism allows any two individuals to mate, but requires complex anatomy. Without preserved reproductive organs, we can't determine which system Wiwaxia used. The energetics of reproduction also remain mysterious. Producing eggs or sperm requires significant energy investment, especially for organisms that broadcast and spawn thousands of gametes. Did Wiwaxia accumulate energy reserves before reproducing, then undergo periods of nutritional stress afterward? Did it reproduce multiple times throughout life, or only once before dying? These patterns profoundly affect population ecology, but leave no direct fossil evidence. One tantalizing possibility is that some Waiwaxia fossils do preserve reproductive information, but we haven't recognized it. Internal structures occasionally visible in exceptional fossils might include gonads, but interpreting dark smudges and organic stains as specific organs is highly speculative. Other fossils show individuals in close association, which some researchers interpret as mating pairs. But these associations could equally represent post-mortem transport, bringing unrelated individuals together, or social aggregations unrelated to reproduction. The reproductive mystery of Waiwaxia extends to many other Cambrian organisms. The fossil record is biased toward hard parts, shells, scales, armour, that preserve well. Soft reproductive tissues, delicate eggs, and microscopic larvae rarely fossilize. This means the reproductive biology of most ancient organisms remains unknown, not just for Waiwaxia, but for the majority of Cambrian fauna. We're left inferring life cycles from modern relatives, making educated guesses based on ecology, and acknowledging how much we simply cannot know. What makes this particularly frustrating is that reproduction is fundamental to understanding evolution. Natural selection acts on heritable variation, which is generated and transmitted through reproduction. Without knowing how Waiwaxia reproduced, we can't fully model how it evolved, how populations responded to environmental pressures, or how it competed with other species. The fossil record gives us beautiful snapshots of ancient morphology, but leaves the dynamic, living processes of reproduction largely invisible. Waiwaxia remains a gorgeously preserved mystery. We can count its scales, measure its spines, examine its feeding structures, but we cannot answer the most basic biological question: how did it make more Waiwaxia? The silence of the fossils on this question reminds us how much of ancient life remains unknowable, locked away by the limitations of preservation and time.